very good evening and a warm welcome to all for another session of Friday Water Thesis Club. The idea of Friday Waters is to wet the Fridays with fun-filled and yet fruitful activities for the future generation. Friday Waters has water talks, book reading, thesis club, and water arts. Focus of the Water Thesis Club is on bringing the student scholars who have done or doing a thesis last research on water matters to the session in order to discuss with them the making of the thesis, research, and the issues around the subject covered. I am Ekta Gupta, the moderator for today's Thesis Club session. I am joined by Professor Sonia Gupta from Watson University as a discussant for today's Thesis Club session. For today's session, we welcome our guest speaker, Mr. Deb Soumya, to share his research on seasonal change in cell size of dominant diatom species in the water of Lake Biwa, Japan. Deb Soumya is currently working as a postdoctoral fellow in the University of Shiga Prefecture, Japan. He has the recent of he was a recipient of one of the prestigious scholarship Japanese government scholarship MEXT in 2018. He completed his PhD from Kyoto University, Japan in March this year. His future goal is to work in the field of polar research and contribute to the betterment of the society. He introduces himself as extremely social and love to live his life in the moment. Welcome again, Mr. Deb Soumya. Now I would like to welcome Professor Sonia to take the session further with him. Thank you. Sonia, over to you.
Yeah, thanks, Ekta. I'm so very sorry. There seems to be a constant issue with my video today. I will be attempting to put on my videos as in the course. But yes, in the meanwhile, Soumya, it is really wonderful to have you here. Your presentation is definitely steeped in scientific discoveries and a lot of experiments. And I really love the fact that you have decided, or you have your motivation is to do some good for the society of today. And wonderful that that scientific, however you're working, can be used for this world, this one world that we have. So with those few words, it is time for you to please share your screen and go ahead with your presentation.
Are the slides visible?
Yes.
Thank you so much. Thank you so much, all three of you. Thank you so much, and it's my pleasure to be here. Once Mansi ma'am told me that can you present in our water conversation, I just said yes. So yeah, I'm very happy. So I'll take it very easy because there are a lot of like scientific terms. Maybe I'll break it down and I'll make it easier to understand. With that, my thesis title was Seasonal Changes in the Cell Size of the Dominant Diatom Species. Let me just... In the waters of Lake Biwa, Japan. And at that time, I was a PhD scholar, PhD student in Kyoto University in the aquatic ecology lab under Professor Shinichi Nakano. And I completed, as Ekta ma'am said, I completed my PhD this year, and now I'm working as the postdoctoral fellow. So I'm completely working different now. I'm not working anything related to diatoms, but I'll present this because this was my PhD thesis. Okay, so what are diatoms? Diatoms are a class of organisms which are microscopic. We can't see with our naked eyes. We have to see under the microscope, and it belongs to Eukaryota, Chromista, Heteroconta, and then Archophyta, and finally Bacillariophyceae. Wait. What are diatoms actually? They are a type of fly phytoplanktons. They are single-celled algae with cell walls composed of silica. And silica are the component of glass. And they are autotrophs. That means they make their own food. And they're primary producers. That means they lie in at the base of the food chain and produces organic matters. The main thing that they does is to convert CO2 from our environment that we exhale. The living organisms exhales and converts it into O2 by the process of photosynthesis. And there are almost about 100,000 species that has been discovered, and there are many more that we still don't know. And they account for almost 20 to 25% of the global carbon fixation. And their main thing, we all know that we all living organisms needs food, and then for them the food are mainly nutrients that are found in the water column of any aquatic environments. And they normally feed on or uptake nitrates, phosphates, iron, and silicate. These four are the very important nutrients for diatoms. They're also ubiquitous in nature, which means they are found in any aquatic environment that persists on Earth. Why is this happening? Okay, so these are diatoms, and the natural sources of water, whether it's ocean, ponds, lakes, streams, rivers, they're found anywhere, and they looks um, exquisite. They're very beautiful, their structures. And by saying that, they have two different kinds of morphologies, which is called centrales, and the other one is pinnales. The centrales are the radially symmetrical, and pinnales are laterally symmetrical. And moving on to the next slide, as you can see, as I said, they're beautiful because when the diatom cell walls are made up of silica, as I told before, and silica is the component, the main component of glass, and that's why they are called the glass house of the aquatic environment or aquatic ecosystem. SiO2 is the chemical composition of glass, and this is an untreated cell with the organic matter present, and this is a treated cell, and you can see that's the skeleton of a diatom, of a central diatom, basically. Okay, and this is the structure of diatom. So I'll say a little bit about the structure of diatom. If you have seen in a chemical lab petri dish, a petri dish is composed of two lids, like one is smaller and the other one that covers it. That is the bigger one. So same in diatoms, there are two valves which exactly represents a petri dish, and the lower valve is called the hypotheca, and the upper valve, which is the bigger one, is called the epitheca. This is the transverse section, this is a griddle view, and this is the valve view. Now, why are they important? Before I say why are they important, I have to tell you about the reproduction or the cell cycle of the diatoms, the life cycle. They have a haplodiplontic life cycle, which means they have two phases in their life. One, the haplo, and the diplo. Haplo means it has haploid cells. That means a cell with one nucleus, and diplontic means diploid cells. So they undergo two kind of reproduction in their life cycle, which is vegetative reproduction, which is mitotic, and it also undergoes sexual reproduction, which is meiosis. And the size reduction occurs due to the vegetative reproduction. So when the cell size reduces, it undergoes vegetative reproduction. But at one point of time, if they continue reducing in size, at some point of time they will perish. So they have to correct this cell reduction and get back to the original size or the initial size of the population, and that happens by size correction, which is called conjugation in pinnate diatoms, and by gamete formation in centric diatoms, and this is the sexual reproduction. As you can see over here, this is very interesting. Centric diatom and pinnate diatoms. As I said, one cell has two valves, the hypotheca, and this is the bigger one, the orange one is the epitheca. So when they divide, the epitheca acts as the epitheca of a new daughter cell, whereas the hypotheca over here doesn't act as hypotheca. It starts acting as an epitheca of another daughter cell and creates a new hypotheca. Like this, the cell size, they keep on dividing, and the hypotheca, which was smaller in size, keeps on acting as epitheca, and hence the whole size of the community keeps on decreasing. So as they divide, they increase in numbers, but reduces in size, as you can see here. Then reaching a certain threshold size limit, they have to correct they have to do some size correction of the whole population, and that's where they undergo sexual reproduction and forms a zygote, and then forms a large specialized structure of cell, which is called an oxospore. And as you can see inside, there is a new cell that's forming, and that's the initial cell, and that is exactly the same size of the parent cell from where the cell division started. And this is the whole life cycle of a diatom. Oh no. Okay. Why is sexual reproduction in diatoms difficult to study in natural systems? Because of the infrequency of the sexual phases, due to the large number of generations, they have a large number of generations, so it's very difficult to exactly and it takes very few minutes and to move on to the next life stage. So sampling from the natural system and then coming back to the laboratory and watching it under the microscope, it's difficult to see the sexual phase. So you have to culture it in the laboratory to actually capture the oxospore stage or the sexual stage, or the cell size reduction stage. Poor profusion of sexual stages, very short generation times, and issues in sexual stage identification and sampling procedures. My aim of the study was first checking the seasonal abundance of dominant diatoms in Lake Biwa, and then the cell size reduction and restoration frequency of those diatoms, and also checking out what are the various factors that are affecting this frequency. What are the uh, mm, environmental clocks that's triggering this kind of sexual stages, life cycle stages in diatoms. My location was in Lake Biwa. It is one of our long-term observation station of Center of Ecological Research of Kyoto University, and the name of the station was IE1. Materials and methods. I sampled for 1 year from June 2020 to June 2021 and collected 24 samples. Conductivity temperature depth profiler was used equipped with an oxygen sensor to measure the profiles of temperature, dissolved oxygen, chlorophyll air concentrations, and also we measured the pH with pH and the Secchi disk, which is used to measure the water transparency or the turbidity, and the nutrients, of course. Now, the sample treatment. First, the samples were collected with Niskinson water sampler, as you can see over here, and it was collected twice. Once with one sample collection, it gives like 6 liters of water. So it was 12 liters of water that was collected and was filtered through a 2 micrometer mesh size and with a 15 centimeter caliber diameter plankton net. As you can see, my colleague is holding it, and we pass the water through it, and we concentrate the samples, and then immediately preserve it in Lugol's iodine so that we can capture the stages. And then after that, we bring back to the sediment to the lab and leave it in the sedimentation chamber for at least a week. And then we concentrate the samples in 10 milliliter vials, and then we observe it under the microscope. The cell abundance was expressed in cells per liter and the cell size in micrometer. And also the cell abundance and cell size were enumerated on the microscope. Now, moving on to the results. First, I'll go through the abiotic factors that we measured. First of all, you can see over here the temperature. So Lake Biwa is considered to be a monomictic lake. Monomictic lake means it undergoes it undergoes winter mixing only once a year. So the whole water column mixes only once a year. As you can see over here, the thermocline is very prominent during summer and autumn and spring. All this time, you can see the surface water is very hot. This is during the summer, which is August through July to September. This is considered as the summer months in Japan. And after that, you can move in through the winter months. You can see over here the water column is very well mixed, and the water temperature is very uniform throughout the water column. The average maximum temperature was 29.74 degrees Celsius, which was in August during the summer, and the average minimum temperature was 8.45 degrees Celsius, which was March 11th, which was recorded on March 11th. Moving on to the pH, during the wet season. So in Japan, it's considered either wet season or dry season because we have rain almost all the time over here in a year. And it's so the maximum pH was 9.11 on June 17th, which is considered as the wet season, and the minimum pH was 7.37, which is in January 21st. Chlorophyll A and water transparency. Chlorophyll A was high during the diatom bloom periods. As we move on to the diatom results, we can see that during this time, which is during the autumn, we have a diatom bloom. And due to that diatom bloom, we have nutrient depletion. Nutrient depletion, I'm saying. We have a bloom of photosynthetic pigments, which is chlorophyll A in the water column, because of the presence of a lot of diatom cells which undergoes photosynthesis during this time. The maximum water transparency was found to be 11 meters, which was during April 21st, which was during the dry season. And during the wet season, the water transparency was very low, 4.5, because due to rain, a lot of water like drains into the basin and the turbidity is very high. Nutrient analysis. We did nutrient analysis, as we said before, that there are four very important nutrients, out of which ammonia, nitrate, and nitrite are considered as DIN, dissolved in organic nitrogen. And we have phosphorus concentrations. These two were determined by the colorimetric methods using the autoanalyzer, as you can see over here in the picture. And silicate concentrations was determined using the spectrophotometer. Now moving on to the nutrient results. Over here you can see during the nutrients, as I told, during this period the water doesn't mix, so you can see at the top the nutrient levels are very low. Over here din is very low, the nitrate concentrations are very low because it has been depleted by the algae that has been growing in the surface water, and due to no mixing, we can see a huge repository of din and in the bottom layers, and it gradually decreases from the top surface water. And over here during the mixing period, this whole repository of nutrient of nitrate or DIN has been brought up to the water column, and it's very well mixed. And the maximum DIN concentration was 50 micromole, and that was recorded during in December 23rd, which was during winter during the mixing period. And the minimum DIN concentration was 15, which was recorded in the surface waters in July 2nd, which was during the summer. For phosphorus, was almost below undetectable levels. We didn't get much of phosphorus because in Lake Biwa, phosphorus level is very low. So nothing prominent was found. Moving on to silicate, as you can see in silicate, silicate is well available in the water column. Over here, silicate is a little more available because during this period before summer, we don't get diatoms. We will see when the diatom bloom actually happens. So during this time, there were no diatoms. That's why silicate was more. And over here, from October to December, this is the main diatom bloom period. That's when a lot of diatoms are found in the water column from top to bottom, and that's why there is a huge depletion of silicate in the water column. And that's because diatom's skeleton is made up of silica, as I told before. So they need to absorb a lot of silicate from the water column, from the environment. So the maximum silicate concentration was 50.66 micromole, which was during October 7th, and the minimum was during the bloom period. By the end of the bloom period in December, that was 9.37 micromole. These are the four beautiful diatoms that I have studied during my PhD work. The first one is Olocosura ambigua japonica, and then Olocosura granulata, and Flagellaria crotonensis, and Prestophanes suzuki. These are the four dominant diatoms that I've found throughout the water column during my sampling period throughout the year. Now moving on to the cell abundance. Now over here you can see, as I told you before, the cell abundance increases with the decrease in the cell size of the population. Over here, ambigua and granulata were dominant during the autumn bloom. As I told you, October to December, October through December is like the main bloom period, as we found in the results. You can see the cell abundance rose to a huge numbers over here just during the bloom period in autumn, and then gradually decreased as soon as the water started mixing or getting colder during the mixing period. That's for Alocasia ambigua and granulata. So follow the red line and the green line, the green and the red. Now moving on to Fragillaria crotonensis. Fragillaria crotonensis revealed two blooms. One was during spring, as you can see from here, from March to June, the turquoise line, and that's the only diatom that was present in the water column. I will show you the pictures in the next slide. And Fragillaria crotonensis single-handedly dominated the whole water column over here. And then it was also present along with Alocasia, granulata, and ambigua during the autumn bloom as well. Over here you can see. And last, we are moving on to Prestophinus suzuki, which was which dominated in winter. So in winter, as you can see, the other three diatoms drastically decreased as soon as the mixing started. But Prestophinus suzuki dominated the winter waters, the cold waters, and remained in the water column until Fragillaria crotonensis appeared in the water column. So one very important thing about Prestophinus suzuki was present in the water column long before, and then it completely disappeared from the water column. And recently, it's been proliferating again, and its numbers have increased a lot. And it's considered as a winter diatom, and that's why it dominated the water column during the cold mixing period. Okay, moving on to the seasonal cell size changes of the dominant diatom. Now moving on to the cell size. As I told you, remember the Alocasia ambigua and granulata was very high during the autumn the autumn bloom. Now over here, the cell size, as you can see, for Alocasia ambigua, the cell size decreased, and it decreased drastically like during the autumn bloom. Same for Alocasia granulata as well, it decreased once. But moving on to Fragillaria crotonensis that had two bloom periods, the cell size decreased twice. And for Prestophinus suzuki, it decreased, and it kept on kept on dividing throughout the winter mixing period, and then it again got back to its original size, or did the cell size restoration, sorry. So Fragillaria crotonensis did the cell size correction twice, whereas the other three diatoms did it once in a year. The lowest average cell size of ambigua and granulata was 20 and 30 micrometer respectively, whereas for Prestophinus suzuki was 11 micrometer. And for Fragillaria crotonensis, it was 68 micrometer during autumn bloom and 75 micrometer during the spring bloom. Now these are the stage pictures of the diatoms during 4 different seasons. In spring, as I told you, it was only Fragillaria crotonensis that single-handedly dominated the whole water column. And then during summer, there were no diatoms present. These are 2 green algae, and that too very less. During the autumn season, the autumn bloom, as you can see, all 4 diatoms are present over here. And in the winter, it's only Prestophinus suzuki that dominated the water column. Now, the relationship between the cell size and the abundance of the species, as I told you, reduction in cell size was proportional to the increase in the abundance for all the dominant diatom species, indicating cell division. All 4 diatom species underwent cell division. Whenever the abundance of their cells increased, they underwent cell divisions. Diatoms in Lake Biwa revealed 2 bloom periods, with Prestophinus suzuki having an extended winter bloom. Now, these are some correlation matrices we did to find out which environmental factors affected or influenced the frequency of the cell division or the cell cycle, cell size reduction and restoration. What we found is, like for Alocasia ambigua, chlorophyll A was positively correlated and to the cell abundance, and water transparency was negatively correlated, whereas for cell size, it was a positive correlation with pH. So pH must have played a very important role in But I would say it's a complex system. It's not only one of them that plays an important role, but it's a complex system that undergoes and times the biological clock of these diatom species. But for Olocosira granulata, it was mainly there were a lot of abiotic factors that affected their cell size restoration and reduction cycle. Positive correlation was found with water temperature, chlorophyll A, negative correlation and conductivity, whereas negative correlation was found with water transparency and nitrogen concentration, DIN, whereas for cell size, there were no significance found. For Flagellaria crotonensis, there was a positive correlation of cell abundance with chlorophyll A and pH, again same as Olocosira ambigua, and there were no significance with the cell size. Now, moving on to Perseverance Suzuki, this was one of the diatoms that I really loved working with because it was present most of the time during the winter season. It was the only one. For the epilimnion means the surface, the positive correlation was found with DIN, and strong negative correlation was found with water temperature, silicate concentration, and conductivity. So this is very satisfying because there are many nutrients that really helped them undergo the cell size reduction and restoration. For cell size, DIN also played a very important role and was positively correlated, whereas negative correlation was found with water temperature and conductivity. Now, we also did RD analysis and of cell abundance and cell size, changes of dominant diatom species with various environmental factors. For cell abundance, the RD analysis explained almost 99.81%. The spring samples clustered together, showing the dominance of Flagellaria crotonensis. As you can see, the spring samples over here, the pinks are all scattered together, clustered together. Whereas for autumn, since the diatoms were of um, different species, that's why they were very much scattered. And also for summer, there were no diatoms present. Moving on to the cell size, the RD analysis explained almost 92.97%, and the samples in autumn, spring, and winter all clustered together because the cell size variation was taking place in all four diatoms. An abundance of Olocosira granulata has been observed in Lake Biwa's water since 1990s, and it is found in moderately eutrophic waters where there are a lot of nutrients available. An increase in nitrate and silicate concentration in Lake Biwa must have been very beneficial. So in 1980s, Lake Biwa underwent eutrophication, where the water became very unhealthy and it was very much polluted because of eutrophication. And then the Shiga government, they decided to lower down the nutrient levels, and that's when they started they implemented some plans, and then the nutrients that was flowing from various catchment areas into the basins were stopped, and also the nutrients were corrected in the basin of Lake Biwa. And that's when the dominant and endemic species in Lake Biwa started disappearing, and then these diatoms, they started blooming more. And that happened to Prestophanes suzuki as well. The dominance of Olocosira ambigua japonica has never been reported before from the waters of Lake Biwa. This is very interesting because this is one of the diatoms that has been reported very recently, and I'm very fortunate to find its dominance in the waters during the autumn bloom. So possibly the temperature, the conductivity, and DIN plays a very crucial role. Well, for diatoms, temperature plays a very crucial role because with higher temperatures, it's the cell processes in diatoms increases more. The silicate concentration depletion after the autumn blooms indicates the high affinity of Olocosira. So Olocosira are known that they are very hungry for silica because they are very long diatoms, chain diatoms, both Olocosira ambigua and granulata. So they tends to take up a lot of silica from their environment waters. And that is why the silicate concentration, as you could see previously in my graph of silica, that it depleted during the autumn bloom. So basically, it's mainly for Alocacea's dominance or bloom. For Flagellaria crotonensis was there in the water of Lake Biwa, but it was not as dominant as it is now. Then that has happened because there was an endemic diatom species of Lake Biwa, which is Olocosura nipponica. Nipponica, by the way, Nippon means Japan in Japanese. So it was an endemic to Japanese water, especially in Lake Biwa, since the 1980s. But it underwent cell size. It also underwent cell size restoration and reduction twice. Um, but Olocosura nipponica started disappearing from the waters of Lake Biwa after the eutrophication was controlled. And because of Olocosura nipponica's disappearance, the nutrients started being more available in the waters. And that's what proliferated new diatoms and other diatom species to take over its position as the dominant diatoms in the water column. So prominent global effects may have played a very important role. The high affinity of nitrate and silicate may have helped proliferating uh, proliferance of Fragillaria crotonensis, and also winter mixing played a very crucial role because it brings up the nutrients from the deeper layers of the water. Conclusion: the cell size reduction and restoration of diatoms by sexual reproduction is very prominent. It got triggered by certain environmental factors, as we could see from our results. There are very different the different nutrients, different environmental factors affected a different diatom species. Not all diatom species depends on the nutrients or environmental factors. And there are still, as I said, it is a complex system. It's a complex process. It not only few nutrients or few environmental factors that affects. So we need more better knowledge about the life cycle of the diatoms inhabiting the lake ecosystem, and also developing future strategies to protect the health of the ecosystem during the climate change. As we know, the environment is getting warmer and warmer day by day. So we need to take care, and for that we need to know more about the organisms inhabiting the ecosystem. Thank you for listening.
I hope I'm audible. My video is still trying to come on. Am I audible?
Yes, sure.
Yes. So, Shoma, thank you so much for your presentation. You have tried to, and I'm sure it's quite clear how you have tried to decipher the scientific terminologies and the concepts that are behind your very, very interesting research. I would like to begin. I know that Mansi is dying to jump in because this is her world, but I will start with some basic questions, and then, of
Sure.
course, Mansi can completely take over. So I was just wondering, Lake Biwa, does it have any locational or environmental definition? I mean, I know that you talked about the fact that Lake Biwa has a history, having the eutrophication that you talked about. Of course, you've written moderately in your presentation. Yes. Would you please, Shoma, would you please stop sharing your screen so that
Oh, sure.
Yeah. Thank you.
Yep.
So, yes, you have touched upon the fact that the Lake Biwa has a history, having the eutrophication, and also you have probably updated us in your presentation. It says moderate, but you have given us a slightly updated picture, perhaps. And you have also talked about the abundance of the granulata and the dominance of the japonica. But I'm still wondering, what made you settle on Lake Biwa for your scientific observations? Yeah.
Okay. Thank you so much for your question. And Lake Biwa is considered as the mother lake of Japan, and it's one of the ancient lakes in Japan. Not in Japan, in the whole world. So, and also like Lake Biwa is one of the biggest freshwater lake. Like I can't see from the other side of the lake to the other side. It's so big, and it provides water to the whole Kinki region, like Osaka to Shiga, Nagoya. It provides water to almost like 3 million people, or maybe 30 million people. Yeah. So a lot of people, and that's why like the Japanese government is very much trying, or they try to keep the lake healthy, because the water, even like it's so clear, the water, you can just drink it like when you go into the lake for like swimming, or when we go sampling, like we can just sample water from like at a certain depth and we can drink. It's
Wow,
that much pure.
fantastic. Yes. Okay. So you chose the history as well as some prevalent conditions for this location advantage.
Yeah,
Tell us that, you have talked about, which is your topic really, reduction of cell size of the diet.
right,
And we see that, in two species, the pH value and the temperature and are correlated. So do
right.
we have a comprehension of why this is happening?
The cell size reduction, I think,
No,
uh,
the correlation between pH value and temperature and DIM.
well, since they're diatoms are very delicate, they tend
Okay.
to react to a very minute change in the ecosystem. And
All right.
diatoms are exactly like the the face of like the ecosystem, how healthy it is, or if the health of the ecosystem is deteriorating. So by that, like if we know that what exactly is affecting the diatom cell size, like we would know that what's affecting the health of the lake ecosystem. So and if you're saying why is it happening, first of all, temperature is one of the key component that affects the um, life cycle of diatoms. So with the increase in temperature, certain temperature, the life cycle gets more active. So temperature affects a lot. pH, of course, it can't sustain in a very acidic condition. So, and nutrients limitation is, of course, the food, it's main. Like without nutrients they won't survive.
Right. So actually, my intention was to understand. You have given us the scientific reasons, but my
Hmm.
intention was to understand what are contributing to these variations. Is there human activity, or is it what is it contributing to that? That was the intention of my.
The human activity, I won't say that there are too much anthropogenic
Right.
effect in the waters of Lake Biwa, because like over here in Japan, it's everything is very controlled. Like it's
Right.
not that someone will just drain something, or some industry will just drain some water. They treat it and then drain it. Like
Okay.
I wish it was the same. I could say for Ganga, that
Good to
flows like very
know.
like 2-3 minutes away from my house. So, but the thing is, like, yeah, I would say like Lake Biwa is very well maintained, and I don't think it's an anthropogenic effect,
Ah, okay.
but it's a sheer effect of the global climate change, especially the temperature. Like the one thing I would say, which is like in winters before, like I'm talking about like what 10 years before, like it used to be like 20 centimeters of snow just in like in the beginning of winter. But now the snow has almost disappeared because of the global warming. I think our climate is warming up, and then last winter we didn't have snow that much. So the climate is changing, so lesser the climate change, lesser the cooling of the temperature, the lesser the cycling of the lake water, and that will affect the health of the ecosystem as well, for sure.
Um, all right. So, um, I we're just wondering that the cell size reduction in abundance. You have given us also the relation between that, but in comparison, what happens when the cell size is reduced? Well, how do these phenomena differ um, than when the same species has a particular cell size? How do their work? How does their presence differ from when the cell size is reduced? What is the difference in the performance? And also, I would just like to, before you answer this question, I would also like to say that yes, so global warming itself has a lot to do, or rather, anthropogenic activity has a lot to do, has a lot of contribution to global warming. So there, I think we have to put that on record also straight. Yes, please go ahead.
Yeah. No. I would really appreciate if you can break down that question a little bit. Like,
Okay, so when I'm
sorry.
what I'm saying is when there you have been, your main topic is the cell size reduction, right?
Right, right.
And so I'm also trying to understand that because I'm trying to always draw it to how humans have affected it, and then how do human beings get affected by it, both
Hmm.
ways.
Hmm.
So when cell size is reduced, what are the processes? Because it is phytoplankton, and we have the very important property of photosynthesis,
Hmm. Hmm.
which we use selfishly for our good.
Hmm.
Now,
Hmm.
how does the change in size of the cells affect these performance that we look forward to?
Well, the reduction of cell size is just a stage of their life cycle. It's not that if they reduce in
Ah,
size, that will affect anything.
right.
So
This is what I'm.
they will undergo cell size reduction and restoration, even if they are living in the healthiest ecosystem. So it's just a
Okay.
part of their life cycle. But
Okay.
like if the health of the ecosystem deteriorates, it can undergo like stunted growth, or maybe it can undergo some disease and they will perish.
Oh, okay.
That's the thing that
Right.
can happen. Also,
Okay.
one thing that I was studying before this, which is like there are infections. There are a lot of fungal infections and bacterial infections that happens when they are not in a very healthy environment. So that's what I was studying before. So in an eutrophic waters, like this kind of thing happens. But
All right.
reducing cell size is not an issue.
It's not an issue anywhere.
Yeah.
Okay. Uh, all right. And the fact that there have been reporting of new species,
Yeah,
right?
it's not a new
So,
species, but it wasn't
or
there
new
before.
reporting.
New reporting.
Sorry, my bad. There's a new reporting of one species which wasn't there, right?
Yeah.
So is that a good thing?
Yeah, it's a very good thing because that means
Wonderful.
the water is healthy. But at the same
Wonderful.
time, there have been, as I told you, like there was an endemic diatom. Okay, let me make this very
Mm,
easy. So presence
okay.
of any diatoms is very healthy, but at certain number. If it's like
Right.
proliferating too much,
Concentration.
that means it has eutrophication. That means the water is not healthy. But Olocosura nipponica, which was the endemic diatom, that was also present, but there were certain sets of abiotic factors that was like helping that diatom to grow. But now the temperature has changed, the phosphorus level has changed, the nutrients have changed, so that is not good for Olocosura nipponica anymore. Now it's helping other diatoms to grow. So that's what is happening right now. So
Okay. Okay. Understood. And that is good to hear. That is very good to hear. So now to come back to what we started with when we had our first conversation, is that
yeah.
how science can be utilized for common good? And how, so I will ask you, how is your research going
Hmm.
to be available to the greater world? And
Mhm.
how will it be utilized for common good?
Okay. So this kind of study was not done in Lake Biwa before. So my professor said, why don't you do this for the like report this from Lake Biwa? Because there has been a lot of work done before, but now but we are moving on to like a lot of advanced tools and everything. But this is like a very first time study. Like this part of this is actually not my thesis, a part of my thesis. So the thing is like first we need to know what is living in the water column, like what's there in the water column, what's affecting their life cycle and everything. So that's why I first started to gather the basic knowledge of the organisms that living in the water. And if we come to know and we have proper knowledge about what's living in the water, what's affecting them, and if the water is healthy, then we can take care of the ecosystem, the health of the ecosystem. And since the aquatic ecosystem is providing water to a lot of people, that means like more this kind of work is done, more this kind of knowledge is gathered to know about the diatoms, the most delicate I forgot the word, it's called uh, well, anyways, like the more we know about these organisms, the more we know about the health of the, or we can keep track of the health of the ecosystem. So this was my first step to knowing the diatoms present in the waters of or waters of Lake Biwa.
Great, wonderful. And do you envision some challenges as you go along with your research?
Challenges now I want to take, like challenges in the sense, well, I did my PhD in a very difficult situation during the COVID. All 3 years of COVID, I did my PhD. So my resources were very limited. So I actually wanted to do a different thing. I wanted to do genetic engineering, but then I had to settle down with this. So I was
Oh,
very much disappointed. But no research is bad. So now what I want to do is like I really want to, as I wrote in my bio, that I want to do some polar research. So I want to learn about the algae under the ice covers, so of Antarctica or Arctic, whatever. So that's what I'm looking forward to. And challenges, I would say there are not many phycologists in Japan. So I want to reach out to phycologists all over the world. Like that's why I went to Hungary. The main reason for me to go to the conference was to get in touch with a lot of phycologists. So to some extent, I did. So yeah, I need the expertise.
all right. And we don't see any obstructions to that happening. So yes, with that, I will ask Mansi. Mansi, please do hop in.
Yes. Thank you so much, Sonia, for taking this conversation and Ekta for actually organizing this event. And yes, Samya, good to see you here again. And yes, I'm usual, always ready with a lot of questions. And I
Yes,
like the word when you used that they are referred as glass house of the water bodies, you know. That's a
yeah,
very simple way to talk about diatoms. The cleaner the glass, the cleaner the lake. As simple as that.
right.
So, when we talk about these, and you also mentioned that they are present in all kinds of natural water systems from ponds to oceans. As a layperson, and I'm always playing that devil's advocate because this is meant for all young scholars and students and those who have no clue about lakes and what life in lakes. Could you please enlighten us if the diatoms are, of course, there are many of classifications of diatoms, but this size thing, does it matter with the size of the water body? It's a very layperson question. Huh? So the pond diatoms and the ocean diatoms, are they friends or relatives, or they are completely different set of diatoms in this range of water bodies?
They are different set of diatoms. Like for lakes, of course, they don't like to live in salt
Yes,
water.
and it's a
That's the
sweet
one that,
water. Yeah, that is
yeah.
exactly why I
But
was
if
questioning.
you
And
see,
flowing.
yeah, if you
Yeah.
see in rivers, I used to work in Sundarbans. Like I worked in
Mm,
Sundarbans for 4 years. So over there, like the phytoplanktons that I used to see in the brackish water, those phytoplanktons, some of them you can find upstream as well as you can find some in the ocean as well, like where the
yes.
water is draining. So they got used to, they evolved in that way that they can live in any saline index. But whereas like the
The
diatoms that you
mangrove
find in fresh
diatoms are
water,
different. The mangrove diatoms are different then.
mangrove diatoms are more steady, I would say.
Yeah, so they can adapt to both kinds of water.
Yes, yes, kind of.
Okay, wonderful.
But for instance, like the freshwater diatoms, they are very different. Now also, if you see, like in freshwater diatoms, let's say like for Lake Biwa, there are a lot of rivers flowing into Lake Biwa. I had one of my paper published, which I worked in the riverbeds. I studied the riverbeds of the diatoms in the riverbeds. They are all sedentary diatoms. Like sedentary, I would say like benthic diatoms. They are
Yes,
attached to the rocks and
much
something.
below in the basin. Yeah,
Yeah, they don't flow with the, because they
yes,
won't stand, because they will be just like
yeah.
into the basin, like flown away into the basin. But the one that are in the lake, they are like free floating, right?
So, so the littoral zone diatoms are more adaptive probably to all kinds of things than the benthic zone diatoms.
Probably,
Yeah.
probably.
Okay. I come to another layperson question here. Because when we look at most of the lake ecologists who are working on these physical aspects of the water system or the physiography, we have always found that algae or diatoms or phytoplanktons, all this family of species, are actually the true indicators of health of lakes, right? Uh, However, when I look at our own Central Pollution Control Board, who is using more than 250 parameters of indicators to talk about the health of the lake, I do not find Diatom sitting there. What is your take on this? We start with pH. Of course, I mean, I can understand that diatoms are some like the smallest module in the lake, which can talk about health of the lake. And what Pollution Control Board and all the reports which we have on the health of the lake, you start with pH, BOD, COD, DO, and all those phosphorus, nitrate, and all those things. And somewhere these living organisms have disappeared in our understanding and monitoring of water bodies in India. Do you see the difference how, especially country like Japan and mother lake like Biwa? I was really happy when you said that Biwa is a mother lake. For many, many reasons, it is considered as mother lake of the world because everybody must know how the lake got revived from such huge eutrophication because it was also the first woman movement in the world to save a water body, the soap movement. So bringing a very dead lake to a live lake where the entire populace around the lake is drinking that water, and then coming to the sophistication of water management in Japan, where they are measuring the smallest module of diatom to understand the health of the lake. Where do you see we are standing in India to really understand lake ecosystem? Because when we measure these other indicators of the lake, are we missing something about health of the lake in India? Because I am really worried because if we are not diagnosing the problem rightly in India, we are not addressing and providing right solutions. What's your take on it? Because you have worked in India also for quite some time.
Yeah. Thank you so much for your question. The main thing that when you said the word indicator, that's what the word I was like thinking. It's called bioindicators, like
I know.
for
Yes.
phytoplanktons, right?
I didn't
Phytoplanktons
want to
are the
interrupt when you were
most
talking.
important
So
sorry,
no, I was saying I didn't want to interrupt when you were looking for that word because I thought you go ahead and then I'll
bioindicators.
come.
Bioindicators was the word like I was
Yeah.
thinking of. Yeah, so not only diatoms, the whole phytoplankton group is
Yes.
the most important bioindicator that's been globally stated. So the
Yes.
thing is, like, I think for Indian waters, like there have been a lot of work actually. I
Mm-hmm.
like NIO Goa or NIO Mumbai. I have worked with NIO Goa and in NIO Goa. So they do a lot of work. Even my Mind Lab also does a lot of work. But I think we need more investment in this kind of studies. And if we invest more, it will like more better papers with more better advanced technologies will be published, and we will have more visibility of this kind of works, which we don't have right now. So I think that's very important, but at the same time, like most of the water bodies in India are polluted, so we know the water is already polluted, so let's just go and measure everything. Is
And just
that
forget about the living creatures on the lake, in the lake.
like almost like most of the lakes like in West Bengal I see like either it's too green or too brown, and they have like in the inner like the Inaka Islam in the rural areas, like most of the lakes have like arsenic pollutions and then those things, right? So probably people are more interested in measuring the chemical contents or this kind of things, the factors of the lake, rather than just studying uh, the living things or the dwelling
The
creatures,
natural systems.
the
Yes,
natural
that's
But
systems, right?
do you think it is also because the way we look at water bodies only from the Anthropocene that we are actually focusing on the other side of it and not looking at lakes as a living entity so that we can look at that there is a life in the lake? My feeling for after studying lakes for 30 years is that what we want to get out of the lake, we are looking for that in the lake. So
Yeah.
we are not really caring about the other creatures in the lake. We just want the water for our appropriation, for human
Mm.
appropriation, and probably that's why we are complacently, or I will say pretending that those creatures do not exist. They are equally living beings as we are. Anyway, so I can keep my thoughts aside. By the way, we also had a very good session on Bhoj wetland from in our Lake Pond series, which we run in Wednesdays for water. And that was also a very nice thesis presentation. It's an ongoing thesis, so I'm going to connect both of you. I'm also going to connect with someone in the Arctic, but they are not phycologists but working on commons, but they can definitely connect you to some people who are working on the Arctic fish. So maybe you can start with fish and get into the diatom through the fish.
Well,
Okay.
I can do
So
the
it's
prey
from the, yeah,
predator
you can do
relationship.
the fish as well. So it's they are from the Ostrom School, so from the Common Studies. So maybe next IASC conference, and you join the early career network of IASC, and you get the whole platform for working on commons and Arctic. They have a serious group there. I need to understand from you one last question here. You know, when you do this kind of specialized study abroad, because I have also done my PhD abroad on something. When I used to do my field work in India, people used to like, they look at my face and governance means what? So they will give me a lot of data on lakes and not really give me data on governance. So, so I don't want to be pessimistic in your case, but I just need to understand because you have worked here also like me before and then went to Japan, and I'm sure you are going to serve the world all over. I still need to understand when we do these kind of specialized PhDs, what kind of scope do we have in India for serious scientists? I'm
Mhm.
sure you are, if you land up in a pollution control board, would you be able to change policies? Or you know that we have a new biodiversity policy, by the way. There is
Mhm.
hardly any mention of diatom, let me tell you that.
Mhm.
Huh? So I see a serious role in scientists like you who can bring a change in policy, the way we look at wetlands and biodiversity, because this is
Right.
part of biodiversity, in my opinion. Where are we standing in India to really bring this kind of change in terms of policy and really start talking about other creatures, smallest of them, and the role of this kind of science in India? Are we taking this kind of science very seriously, or they are just a niche for very good publications? I know biotech and biology and geology have very high peer-reviewed journals and publications. In practice, where do we stand?
To be very honest, I feel like, as you said, like, will you be able to change? I don't know if I will be able to change, but I can definitely voice. Like,
Yes,
it's not my own, like, I
of
can't do
course.
it single-handedly, but I
Of
can
course.
preach people. And like, I would say, like, since we are a country with 1.2 billion people, now it's
Or
1.4. Now it's 1.4.
yes.
Yes, I corrected. I
The
corrected.
more we grow, the less the diatoms are in the lakes and ponds and rivers. So that's my problem.
So yeah, we all have to work together. And like, the thing is, like, everyone has to be very much responsible for what they do in their daily life and contribute very responsibly. They don't have to do everything for the whole country. At least they do good for themselves. If everyone does good for
Yeah,
themselves,
they're nearby Pukur and Lake, huh? If they can do good
right,
for them. I'm living
right.
in Kolkata right now, by the way.
Yeah, that's the thing I feel like. And that comes with knowledge, and that comes with more telling and preaching. And the people nowadays, like, they're so busy with life, these things happens very less. Like, they're only like doing research in their laboratory, they're publishing papers. But actually, like, what you guys are doing, like this kind of research, I wish there were more people to know about what we are all talking about. That would have spread more words, more knowledge. And knowledge is
We
power, of
will
course.
spread your work. We will spread, and that we can promise you from here, because we actually, it's an open education platform which we are creating, and we are getting serious scientists like you people and just
Thank
making
you.
a repository of knowledge here. So I will rest my last question here. If Ekta, are you fine? Last question, please.
Of course.
Hmm.
Yes, please.
Can you give us an alarming picture with keeping climate change in mind? Um, we know that more diatoms means reaching towards eutrophication. But total full presence of diatom or overpopulation of diatom, just like human beings, is equally serious as total absence of diatom, right? Keeping climate change in mind, I would like to know from you, are there studies where people have come to know that diatoms are highly adaptive creatures to this changing weather, to these pollutions which the Anthropocene is forcing on them? How adaptive these species are? Because you did talk about the sexualism of theirs, you know. So, what is your take on this, and what kind of studies, and what are studies telling us about it? Are they going to adapt? Because I'm very keen and curious all the time nowadays, which species or which species are going to survive this whole wrath of climate change. I hope humans don't, but I may sound very pessimistic, but I really feel that we should not survive it because we have screwed it up.
The
Because I
pace with
think that
which we are.
the minutest or the module or the smallest module of any unit are the most resilient. So I just need to know how resilient and adaptive the diatoms are. Yes.
Thank you so much. I would say diatoms are not at all resilient or adaptive. As I
Okay.
said, like from my presentation, Olgosaura niponica
Yes.
completely perished from the water. So it's like that. So since they're very delicate, they don't survive that much changes. So it's just that they will go, but then like the one that can survive will take its place. It's not that the same thing that will evolve to or get adapted to the new environment. So
Interesting.
that happens with them.
Yeah, because there was some relationship which was established between dinosaurs and birds and all those things, you know. So it's interesting to learn that. I'm done for now, but we can keep this conversation going once you are on board our W4W family. And over to you, Ekta. Thank you so much, Soumya, for this candid conversation. Yeah, thanks.
Thank you so much. My pleasure.
Yeah. Thank you, Mansi. Um, Thank you, Soumya, for the wonderful presentation and beautiful insights and eye-opening. It's basically so. We always, in every session, I feel that we are discussing the things that are visible. Even we are ignoring the things that are visible to us. And today we were talking about the microorganisms, so which are not even visible. So how much responsible we are. So this is the actual thing. I feel that whether we are responsible for ourselves also.
I
So
think
because we are,
sorry.
yes, please. Yes,
Um,
please.
I think those are still organisms. If we put them under the microscope, we can still see them. But then we have air that we can't even see, just feel. So we should be responsible for everything that's around us. So
Yes.
Very well
In
said.
every
Yes.
section, only one thing that I feel prominently, that we are not alone on this planet, but we live like this, that we are alone on this planet, and we are not even able to realize. We don't want to realize. Even we do, but
mm,
we don't want to realize that we can't survive alone. So this
right.
is the whole, yeah.
I
So
went back to India after, I'm so sorry. I just a small thing. I went back to India after 4 years, and I was like I felt like in Kolkata, as I see, the air index is very bad right now. So,
Yes,
and it's
very
very much
bad.
polluted. So, in Japan, we can't throw anything on the streets, right? We have our designated places where we should throw. So, literally, when I was out with my friends, they were just throwing stuffs, and I literally collected in front of them. I know they teased me. They told me, "Oh, this is not Japan and whatever." But I said, "This is where we live." So, if in your room also you make it dirty, it will be difficult for you to stay, right? So it's same.
That's the
Like
way.
locality is your room, is your whole environment. So you should take care of it. So these are
Kudos to
small
you,
things.
Shobhit.
exact,
Kudos
Yeah,
to
yeah.
you.
yes,
That's
yes,
that's the way ahead. Absolutely. Yes.
Even like after having
exactly.
the cigarette, like people are just throwing, my friends are just throwing. Even I also was smoking, and then I took them and put it and brought it to my home and throw it in my trash. So these things, maybe out of five of my friends, one of my friends will see, and they will also start like
Right.
this.
Yes.
This is how we
Even
spread the responsibility, the sense of responsibility, I would say.
right.
Yeah, Soumya, the same thing that I would like to share as you have shared. Today I went to the market and they were packing the, the groceries in the packets and all. I said, please don't give me this plastic bag. No, no, ma'am. The owner will scold me for this. So when finally they were packing all the grocery in the polythene bag, a lot of people were there. They were asking, give me a packet, give me a packet, give me a packet. So I said, in front of them, I packed all my things in the jute bag. So maybe at any moment of time, they will realize that we have refused for the plastic bag. So
Right.
that's what we can do only.
So I go a
Yeah.
step ahead.
So
I tell the shopkeeper, please stop stocking plastic bags. I'm
Reducing
going to
the
come back
consumption
tomorrow
will
and check on
immediately
you.
exactly. You know, shopkeeper can't stop this. When we do these things in front of a lot of people, when we are purchasing the things, I think that may change if we are doing it in front of the 10, 20, 50 people. So if one, even single person can change their lifestyle
Yes.
by
yes.
seeing this thing that somebody has refused to take the plastic bag. So maybe it can be a chain reaction.
Yeah, the
So
charity
with
charity
this,
starts at home, no?
yeah, exactly. So it's a wonderful session, and definitely thanks to Mansi for organizing these wonderful sessions. And thank you, Sonia, for accompanying us as a discussant and taking out the best from the speakers and their research. So it's a really very knowledgeable sessions, and looking forward to organize many more sessions. So with the Soumya, looking forward, as Mansi said at the start, yeah, refer more research and the speakers for our sessions. So it's time to say goodbye, but not the bye forever. We will be meeting soon on the. It's for all the viewers, whether they are watching now or watching live and later on watch these sessions. Join us on every fourth Friday to every third Friday, I think. I missed it. It's third Friday at 5 p.m. to learn more on the water researches from more speakers. So join us for the next Friday session, the forthcoming Wednesday for water session at 5 p.m. Do join on biodiversity series. So watch us on www.w4wfoundation.in. Thank you for joining us and watching us. Thank you.